Good day, this is Professor Will, CBMD PhD. Today is Thursday, August 20th, 2020. It is 4:31 and 48 seconds a.m. Eastern Standard Time. This is a continuation of my corona research. But before we get started, let's take a look at something I wrote. In today's video diary, I would like to talk about my progress on my ongoing research and projects. To start, there has been a bit of a gap in my corresponding research during summer months. The reason being, perpetuating into summertime, I spent time taking long walks, which has been making me extremely tired. A lot of sleep, work, and exercise, including my past housekeeping job. Moreover, transitioning into the fall season, I plan on working on that microscope prototype I visualized and designed in my laboratory, and congruence to my projects. In today's perspective, I plan on finding a job established in computers to hopefully finance my projects. In addition to shoveling driveways in the winter to barter food, excited and accomplished about my new goals. Furthermore, in today's short video diary, I would like to announce that during this periodization, I'm feeling excellent and confident to continue with my goal sets and progress. Well, I hope my audience enjoyed my presentation. Hope to see you again. Until next time, let's continue on with my corona research. Toward the coronavirus breathalyzer test. Date August 19, 2020. Source American Chemical Society. Summary. Researchers have developed a prototype device that non-invasively detected COVID-19 in the exhaled breath of infected patients. Few people who have undergone nasopharyngeal swabs for coronaviruses testing would describe it as a pleasant experience. The procedure involves sticking a long swab up the nose to collect a sample from the back of the nose and throat, which is then analyzed for SARS-CoV-2 RNA by reverse transcription polymerase chain reaction, RT-PCR. Now researchers reporting in ACS Nano have developed a prototype device that non-invasively detected COVID-19 in the exhaled breath of infected patients. In addition to being uncomfortable, the current gold standard for COVID-19 testing requires RT-PCR, a time-consuming laboratory procedure. Because of backlogs, obtaining a result can take several days. To reduce transmission and mortality rates, healthcare systems need quick, inexpensive, and easy-to-use tests. Researchers wanted to develop a non-material-based sensor that could detect COVID-19 in exhaled breath, similar to a breathalyzer test for alcohol intoxication. Previous studies have shown that viruses and the cells they infect emit volatile organic compounds, VOCs, that can be exhaled in the breath. The researchers made an array of gold nanoparticles linked to molecules that are sensitive to various VOCs. When VOCs interact with the molecules on a nanoparticle, the electrical resistance changes. The researchers trained the sensor to detect COVID-19 by using machine learning to compare the pattern of electrical resistance signals obtained from the breath of 49 confirmed COVID-19 patients with those from 58 healthy controls and 33 non-COVID lung infection patients in Wuhan, China. Each study participant blew into the device for 2 to 3 seconds from a distance of 1 to 2 centimeters. Once machine learning identified a potential COVID-19 signature, the team tested the accuracy of the device on a subset of participants. In the test set, the device showed 76% accuracy in distinguishing COVID-19 cases from controls and 95% accuracy in discriminating COVID-19 cases from the lung infections. The sensor can also distinguish with 88% accuracy between sick and recovered COVID-19 patients. Although the test needs to be validated in more patients, it could be useful for screening large populations to determine which individuals need further testing, the researchers say. Is the COVID-19 virus pathogenic because it depletes specific host microRNAs? Date August 14, 2020. Source University of Alabama at Birmingham. Summary. Researchers propose that the COVID-19 virus acts as a microRNA sponge to deplete miRNA levels in ways that aid viral replication and stymie the host immune response. Why is the COVID-19 virus deadly? While many other coronaviruses are fairly innocuous and just cause colds, a team of University of Alabama at Birmingham and Polish researchers proposed an answer. The COVID-19 virus acts as a microRNA sponge. This action modulates host microRNA levels in ways that aid viral replication and stymies the host immune response. This testable hypothesis results from analysis of current literature and a bioinformatics study of the COVID-19 virus and 6 other coronaviruses. Human microRNAs, or miRNAs, are short non-coding RNAs with about 22 bases. They act to regulate gene expression by their complementary pairing with specific messenger RNAs of the cell. That pairing silences the messenger RNA, preventing it from being translated into a protein. Thus, miRNAs are a fine-tuned controller of cell metabolism or the cell's response to stress and adverse challenges, like infection by a virus. The miRNAs are only about 0.1% of total human cell and tissue RNA, while replicating viral RNA of a virus like the COVID-19 virus may reach 50% of the total cellular RNA. So, the UAB and Polish researchers say, if the COVID-19 virus has binding sites for specific miRNAs, and these sites are different from the binding sites for miRNAs found on coronaviruses that cause colds, the more pathogenic COVID-19 virus may selectively sponge up certain miRNAs to dysregulate the cell in ways that make it a dangerous human coronavirus. Sponge idea is not novel. Viral RNA sponges have been shown capable of removing host miRNA by the Epstein-Barr virus, and sponge activity has also been shown for the herpes and hepatitis C viruses. There were 2 human coronaviruses prior to the COVID-19 virus, whose formal name is SARS-CoV-2, that foreshadowed the devastating consequences of the COVID-19 virus. The first was the severe acute respiratory coronavirus, or SARS virus. In 2002, the second was the Middle East Respiratory Syndrome coronavirus, or MERS virus in 2012. Neither had the high infectivity of the COVID-19 virus, but both were dangerous, causing 774 and 866 deaths, respectively, according to the National Institutes of Health. In the present study, the researchers used computer-aided bioinformatic analysis to find potential miRNA target sites for 896 mature human miRNA sequences on 7 different coronavirus genomes. These genomes included the 3 pathogenic coronaviruses, the SARS, MERS, and COVID-19 viruses, and 4 non-pathogenic coronaviruses. The researchers found that the number of target sites was elevated in the pathogenic viruses compared to the non-pathogenic strains. Furthermore, they found that pathogenic human coronaviruses attracted sets of miRNAs that differ from the non-pathogenic human coronaviruses. In particular, a set of 28 miRNAs were unique for COVID-19 virus. The SARS and MERS viruses had their own unique sets of 21 and 24 miRNAs, respectively. Focusing on the 28 unique miRNAs for COVID-19 virus, the researchers found that the majority of these miRNAs are well expressed in bronchial epithelial cells, and their dysregulation has been reported in human lung pathologies that include lung cancer, chronic obstructive pulmonary disease, cystic fibrosis, and tuberculosis. Furthermore, many of the miRNAs have been proposed to act as tumor suppressors that target the pathways for programmed cell death, or apoptosis, that are supposed to make a cell kill itself when infected, mutated, or stressed in other ways. Reduction of those miRNAs have been associated with poor cancer prognosis. Hence, the COVID-19 virus, by its potential reduction of the host miRNA pool, may promote infected cell survival and thus continuity of its replication cycle. The authors gave a detailed explanation of how the virus replicates inside an infected cell, including how the cell assists protein folding and how the virus begins assembling the cell's endoplasmic reticulum and Golgi system. They also describe many of the cellular proteins involved in these steps. This viral replication is known to reduce stress and can provoke an unfolded protein response that causes a cell to undergo programmed death. Taken together, the researchers said, the viral strategies to increase the endoplasmic reticulum, membranes, and endoplasmic reticulum folding capacity, and block unfolded protein response associated translational attenuation. Inflammatory responses and apoptosis are critical components for virus production. The authors then showed, by citing literature, that 9 of the specific cellular miRNAs that potentially are sponged by the COVID-19 virus could help achieve those viral needs. The host miRNAs, potentially controlled by the pathogenic human coronaviruses, may be the key to gaining control over a very limited and specific set of miRNA targets. The researchers used computer-assisted gene ontology programs to find the genes and cellular pathways affected by the pathogenic human coronaviruses, and by the COVID-19 virus in particular. The pathways they found further supports the hypothesis that pathogenic human coronaviruses, including the COVID-19 virus, utilize the host miRNAs to adjust cellular processes in order to facilitate their viral protein production. Our hypothesis will require validation, starting with the assessment of these miRNA levels in infected tissues, and ending with restoring the host miRNA balance with miRNA analogs. Furthermore, completely understanding how viruses take advantage of the endoplasmic reticulum and unfolded protein response pathway may also lead to the novel therapeutic strategy. One is the varying susceptibilities to infection seen among patients, including a more severe morbidity and mortality for older patients. There may be individual differences among patient miRNA profiles. A recent study has suggested that COVID-19 virulence in AIDS patients may be due to a lower abundance of mRNAs, and this may be a contributing factor in disease severity. Another biological question is how the virus coexists in its normal animal source, bats. Notably, the researchers said a recent study proposed that bats, considered as host of origin for the COVID-19 virus, have tolerance to potentially deadly viruses because of specific miRNAs. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.